I would now like to welcome to the floor our next speaker, Ilan Samish, the founder and CEO of Amai Proteins, who launched the company after publishing the world's leading book on computational protein design. He's also a graduate of the Tel Aviv University Interdisciplinary Program for Fostering Excellence, and has a PhD from the Weizmann Institute. Did his postdoc at the University of Pennsylvania, and since then has led a very rich academic career with a focus on protein science, and as a lecturer in genetics, biochemistry, physical chemistry, bioinformatics, and computational biology. Today, he's joining us to speak on the topic of the sustainable future of precision fermentation sweet and other alternative proteins. Ilan, it's a pleasure to have you with us. The floor is yours.
Thank you, Franco. Really appreciate it. And thank you for the invitation. So I'm happy to talk about the sustainable future of precision fermentation, sweet and other proteins, focusing on designer proteins, but this will be only one part of my talk. I'm also a member of the FoodTech committee of the Israeli Fermentation Society. I wanted here not to give you the regular well-polished pitch about Amai, but to give something which is more on the trends of the field of precision fermentation. So this is just a collection of slides from here and there and not the usual polished pitch. But before I begin, I would like to congratulate Deep Knowledge Analytics for doing an amazing job in their briefing and analysis of the FoodTech landscape. And within this, following what already Yuval said, that there was a good representation of the Israeli FoodTech scene, they have noticed that Israel has 12.5% of all the top FoodTech firms. And this is in a country that is very small. So if you compare this to the size of the population, by far Israel has briefly an order of magnitude more startups than any other place. And if you want to hear more about it, just for the fun of it, I did write at one time the Startup Nation rap, and you're welcome to listen to it. I will share it in the chat later on. So let's move to substance. And we're talking about food, and let's talk about cheese. And cheese is something we all take for granted. However, in the past, in order to make cheese, you thought about different animals helping it. And many of us thought about mice, including some kids who think that the mice are the ones doing the holes. And actually, to make cheese in the past, you took milk and you threw into it the belly of usually a pig, because you needed an enzyme called chymosin, rennet. And today, this enzyme, 99% of the cheese in the world is produced by an enzyme that is made by precision fermentation. It is made by E. coli, Aspergillus, yeast, or other organisms, and we don't know about it, because this is what is called a processing agent. This is something that is part of the enzyme industry, which is a huge industry of the white biotechnology, the commodity biotechnology, which is different than the pharmaceutical, the red biotechnology, or the green agriculture biotechnology, or the blue aqua biotechnology. And as the Rethink X very good report has outlined, the world is moving to making more and more proteins by precision fermentation. And the reason for that is very simple. Number one, we cannot sustain the world without doing it. Already now we don't have enough space for all the cows, and it's not sustainable also because of so many other reasons. And also the efficiency is not so good. The feedstock efficiency is 4%, while the efficiency of precision fermentation is 40 to 80%, according to this very good report. And here there is an example of the Pichia pastoris, which is used to make leg hemoglobin, which is done by Impossible Foods. And if you move from well over 100 different proteins which are made by precision fermentation as processing agents, namely they are not part of the label, but part of the making of the protein, and you move from there to proteins which are actually making a part of the ingredient. Today, you only have 3 such things. You have the eye structuring protein of Unilever, which is actually no longer done. This was done to help mitigate small ice droplets within ice cream. You have the leg hemoglobin of Impossible Foods. You have the beta-lactoglobulin of Perfect Day Foods in filamentous fungi, and hopefully we will be next. There are some other companies that are now in the process of doing so. And just a word about AMI. This talk is not about AMI, but part of it I'll just use as an example. AMI is about fixing our food system 1 protein at a time by combining computational protein design with precision fermentation. Our first product that is going to the market is the world's sweetest and most stable protein. And we have in our pipeline specific proteins for the meat, plant, and milk space in order to make products which are tastier, more stable, more cost-effective, digestible, hypoallergenic, and produce them in a sustainable, environmentally friendly way. And we do that by combining several technologies. The first one is called computational protein design, and I'm happy to send my book on the field to anyone interested. And the second one is called precision fermentation. So let's talk a little bit about these 2 technologies. The first one is computational protein design, and why do we need to redesign what is already found in nature? And the reason for that is that proteins are intrinsically unstable, and proteins live in their natural habitat, in their small piece of heaven. Whether heaven is our body, whether it is an exotic fruit in the jungle where you have sweet proteins. However, the mass food market is in extremely harsh conditions for a protein. You need long shelf life, you need very high concentration, you don't have a buffer, and you need to produce it in a very cheap way. And for that, we need to learn from proteins which do live on the edge. And you have life on the edge. You have life in the Dead Sea. You have life in hot springs deep in the ocean, in acidic swamps, and so on. And by mimicking proteins which live in such conditions, using computational protein design and combining them with technologies, with functionalities which are good for us and found in regular proteins, you can make new designer proteins which will really be fit for the mass food market. And then in order to produce them in a sustainable and cheap way, you use precision fermentation. Fermentation is actually the oldest technology in the book. Beer, wine, kimchi, yogurt, in all of these cases, you use fermentation, and at the end of the day, you eat everything that is in the fermenter. In precision fermentation, you add a layer of harvesting, and you harvest from the fermenter the white powder. And at the end of the day, our business is to sell white powder that makes people happy. So we have proteins for the plant space, for the milk space, but we are most known for the protein, which is the first healthy, tasty, cost-efficient, and sustainable sweet protein, which can replace sugar in a very nice way. So just to sum up the pipeline, we begin by redesigning or designing novel proteins, which are, of course, proprietary. Then we grow them from a Petri dish to a large scale, and then we make food. These are some of the different types of foods that we have made, and there are many others which are still not under patent, and consequently we cannot show them. And what we do is we do not compete with the diet market. We actually compete with the sugar market, which is much bigger. So in each product, we reduce the level of sugar to a level where the average customer will not be able to distinguish between the full sugar product and our product. In ketchup, for example, this is 70% reduction of the added sugar. We are good for liquids, for solids such as dark chocolate, for hot applications such as a hot cup of tea, and also for alcoholic and non-alcoholic applications. We have numerous collaborations, PepsiCo, Danone, Ocean Spray, and several others that we are not allowed to disclose. And in order to make sure that what we have is really good, we have a professional super taster panel, mainly students, very well screened, only the super tasters that taste what we do in a blind way with a statistician following up on what we are doing. So to do that, we have what we call the Pro Protein platform, which is pro people, pro taste, pro planet, pro health, and pro industry. So let's talk a little bit about the industry and this huge revolution of moving to precision fermentation. What do you need to do and what is out there? So first of all, in order to get good technoeconomics, you need high yield expression, which is how much time, how much money, how much grams per liter you have, what is the quality, how much energy you need to put into the purification of the protein. And for that, you can do it yourself. We have numerous collaborations also with academia. There are numerous vendors that can help you to do that. And the idea is that your factory becomes a brewery. And just like brewery and beer breweries are all over the world, the only difference is that you add oxygen because you don't want alcohol, and you add a layer of harvesting because you want a pure protein. And if you have a pure protein, from the regulatory standpoint, it is GMO free. You don't need to go through the GMO tracks. And for that, just to take some of the considerations, you want to be free of antibiotics. There are some ways to do that, because in many places you cannot do large-scale growth with antibiotics. CRISPR today is a very easy and convenient way to remove the antibiotic cloning resistance post-cloning. And you want to purify the protein. Of course, you have to go through FDA, so you don't want toxins and other things. And you want also to use purification tools that are very cheap and do not contaminate. In academia, you use a lot of affinity chromatography with a nickel column, which is not good. You basically really want only to filter things using membranes, tangential flow filtration, ultrafiltration, and so on. It's also important to have a cheap media as to the growth, and this is today in the alternative proteins for the meat. This is one of the big things to optimize the media cost. And you want also to purify the protein to a good level, but not more than that, because food is not pharma. And in pharma, you need a very, very pure protein. For example, in Impossible Foods, according to the GRAS application, they have a purity which is less than 70%, because you can simply eat all the other proteins that come out of the yeast where they make it. So we spoke about the purification methods, and let's talk about one of the biggest issues, and that is consumer approval. Just like with any revolution, you have today something, and we saw it with the COVID-19, where you need to have consumers who love what you do. And today there is a bad name for GMO. In our case, we are GMO free, but this is an issue which is more religious than science. And you need to show that the product does not contain DNA. Most people don't know that when they eat a tomato, they eat DNA. And that's another important thing. And you want to be natural, but it is very difficult to define what is natural, because at the end of the day, you have 100% protein. Even if it is a novel sequence, it's just like any other protein, a necklace of beads called amino acids. And on that, you have discussions, and it's actually a new world that we are moving into. And then you go, what is exactly novel? For example, the perfect day foods that are using the beta-lactoglobulin of milk, they actually have a patent over the different glycosylation pattern, which is post-translation of the protein. So it's actually a slightly different molecule, but it's all done by biology. So it's made by biology. Is it natural or not? I leave you with a question. You need to answer it. So for that, a year and a half ago, the FDA has decided that they need to get ready for the next revolution, and they made the Institute of Science and Global Policy made a meeting of 56 stakeholders. We have caucuses every day. We had only 2 startups there. There's a CEO of Perfect Day Foods and myself. They spoke about the diversified protein future. I spoke about the language of Food 2.0. And I think that we need, as scientists, to move from being science-centered to client-centered, just like with hospitals. You move from hospital of the sick to health center because these are your clients, and you don't want to engineer proteins, you want to design them, you don't want to do recombinant proteins, you want to do heterologous expression, you don't want to mutate, you want to substitute. These are all important terms where you want to really have your product being embraced by the client who is a consumer that wants all the knowledge, but is often puzzled by some of the scientific jargon. And just how you call fermentation-derived food, there is a lot of different names. Flora-based, as Perfect Day Foods call it, lab-made, culture, synthetic. Those are all names which there is still a dilemma about, and actually SynBioBeta, the biggest industrial synthetic biology meeting, is moving now to a new name, which is made by biology. So I have a paper about this field, and I think this is a challenge we all need to address as we go to the market about the public relations, a misunderstanding, and to get a better consumer acceptance about this disruptive innovation, which at the end of the day is really good for the consumer as it is sustainable. And this is not different than any other big scientific revolution. And we can discuss a philosophy about that, but we don't have time to do that. We should also discuss a sustainability aspect. And there is a question, what is the alternative? And you also need to understand the waste management, because at the end of the day, you have yeast, for example, or filamentous fungi that made your protein, and how do you handle it? In some places you have an issue because this is called a GMO and you cannot work with it. So the waste management is part of what you need to take into account. Where and what are the rules of this GMO waste? And you also need to take into account that today there is excess food grade fermentation capacity in the industry, but it may need some modifications, but in the future, it seems like we will need much more large-scale food-grade fermentation capacity because this field is totally exploding. In the case of AMI, we have 2 tracks of sustainability. One is a health track. The other one is the fact that sugar overconsumption contaminates air, land, and sea and affects the lower socioeconomic class the most. And even an investor banks such as Credit Suisse said that without sugar tax, our World Health System will collapse. So just to finalize, you also, of course, have the economics, and actually you have to look on competition and trade secrets and on the IP and on the economy. In our case, we are actually cheaper than sugar in sweetness units because our protein is 4,000 times sweeter than sugar, meaning that we replace a kilogram of our protein is worth 4 tons of sugar, or we replace a teaspoon of sugar with just 1 milligram of our proteins. I can go a lot about the issue of sugar overconsumption and how the 8 different categories fail in taste, cost, health, and product fitness. And consequently, most of the market is still not the diet market, but rather the sugar and high fructose corn syrup. And even these, whether it's sugar or not, those are small molecules which do have adverse effects. In our case, we use big molecules, proteins, which build our body muscles and tissue. They activate the sweet receptor just like any sweetener, but then they are digested to their amino acid constituents, and consequently, they are healthy, and we have all the digestibility data in order to prove this. So just to sum up, we are taking proteins found in plants in the jungle. We never saw the plant, we never saw the protein, we just looked at their 3D structure, and these are very good. However, they suffer from cost stability and the suboptimal sweetness profile. So using these as templates, we redesigned the protein in order to get a cheaper protein, a higher stability one, and one with an optimal sweetness profile. We have won numerous awards along the years. We are active since 2016, and we have an amazing pool of investors and a very strong board of directors. We are now in the final stages of approval within the FDA, and we are having next month a official meeting with them. And just to sum up my part, what we do with this, we take the huge challenge of sugar overconsumption with the breakthrough technologies of precision fermentation and agile integrated computational protein design to present a radical solution, which is a first sustainable, healthy and tasty protein. And actually, sugar overconsumption is also affecting COVID-19, and people who have insulin resistance simply have more gates for the COVID to get in, and consequently they're more prone to have more serious COVID-19. And just as with COVID-19, we understood that we need preventive medicine of vaccination. Likewise, today the biggest health challenge of a non-communicable disease is sugar overconsumption, and this is a huge burden on quality of life and also on economics. So after success in curing disease, it's time to cure the food we eat. And for that, you need to design proteins which are fit to the mass food market and produce them in a sustainable and cost-efficient way, which is precision fermentation. Just as an example, stevia produced by precision fermentation is half the cost of stevia produced by agriculture. So thank you very much, and I'm happy to go for any questions.
Thank you so much, Ilan. That was illuminating. For anybody who has specific questions, please feel free to pop those into the Q&A box at the bottom of the screen. We'll do our best to account for those during the panel discussion Q&A a little bit later.